All right. I'm really sorry for the delay due to technical minor problems. We can now start, and we are live and recorded. So thank you very much, Marc, for joining us, and everyone in the audience. I am pleased to introduce Marc, who's kindly approached us and suggested his contribution to the course and Ukrainian bioinformatics community. So Marc is professor in Norway University of Bergen and is also affiliated with Norwegian Institute of Public Health and Computational Biology Unit. His group is using multidisciplinary approach on large cohorts to get new knowledge about metabolic health of early childhood and during pregnancy. And Marc has also contributed to various proteomics tools, which are widely used in the community. And today he'll talk about his current research. The mic is yours.
Thank you very much. It's a pleasure to be here. I will talk today about a kind of emerging branch of proteomics and its use in genetic epidemiology studies. And as said during your kind introduction, I'm working in Bergen at the Mono Research Center for Diabetes Precision Medicine, where we work on metabolic disorders in early childhood and in pregnancy. And one of the questions that we have been tackling the past few years is, why are mothers sick in early pregnancy? If you watch a series, you would typically see someone gets pregnant or they start vomiting, but there are much more symptoms than that appearing in early pregnancy. And while in the culture these things are trivialized and we say, yeah, everyone is sick in early pregnancy, you really have a wide spectrum of what symptoms are appearing and how strong they can be. In the worst case, nausea and vomiting in early pregnancy can be a threat to the life of the mother and the baby. And with that, I can show you an example from the UK, where in 2022 in The Guardian, health professionals were saying we need to take women seriously when pregnancy sickness gets too strong. They need help. And you see 2 years later, Jess Cronshaw, who has been extremely sick in early pregnancy, has lost the battle and died from not receiving proper help, and her baby died as well. And during years and years, people have been wondering why women are sick and looking at hormones that follow the pattern of nausea and vomiting during pregnancy and just looking at the wrong direction. Until a few years back, where someone decided to take more of a genetic approach and to look at whether we can see patterns in the genome that associate with nausea and vomiting during pregnancy. And so basically screening the whole genome, check whether sometimes if you have a change of letter at a specific place, the mothers will be more sick. And this has been a seminal paper in the field led by Marlena Fejo, where you see in essence here all the chromosomes and all the small changes of letters that make us who we are. And here a huge peak next to the gene GDF15. And later on, more research has definitely shown the link between GDF15 and pregnancy, and especially this work with Marlena and led by Steve O'Reilly, that really demonstrated how GDF15 triggers the symptom, and it's not necessarily GDF15 itself, but the maternal sensitivity to GDF15. And for all her work in the field, Marlena has been, you might have seen it, been awarded Woman of the Year, I think it was in 2024. So it's not every day that a scientist gets Woman of the Year, and that is quite an achievement. And so this is a textbook example of what we call genetic epidemiology, the analysis of health and diseases in populations through the lens of genetics. And here, once again, we're really looking at things that happen population-wide to try and understand the common determinants of diseases. So I said we are looking at genetic variation, the small changes that make us who we are. And you can stratify, if you like, genetic variation depending on whether it is germline or somatic, which germline would be what you inherit from your parents, whereas somatic happens to you after conception. And most of the time in genetic epidemiology studies, we focus on germline variation. You can also look at genetic variation in terms of small changes in the sequence or broader changes over whole stretches of your genome. This is sequence variation versus structural. And in genetic epidemiology, very often we focus on sequence variation, so changes of letter, and typically changes that are rather small, not too large. And once again, we focus on things that are common in the population, not too much on rare symptoms or if you have like one mutation that would give you cancer, one mutation that would give you a syndrome. That's not necessarily what we're going to talk about today. And you can think the link between something being common and pathogenicity. That's something that we call the genetic architecture of diseases. If you have something that is very common in the genome, it's not likely to make you sick. So we are not looking at things that are going to have a big effect on your health, but common changes that show patterns that are true over a whole population. A typical plot that you would see in this type of studies shows the effect size against the minor allele frequency, so how often the rare event in the genome happens. And you see here it's an analysis of height. The genes that are most associated with height in this type of studies, you would find them in 20% of the chromosomes in a population. And when we talk about strong effect here, you see it's half a centimeter difference. So if you have one allele versus the other, you will be half a centimeter taller or smaller. It's not much, but it's something true over the whole population. And this helps us putting the finger on some genes that are essential in the development of a trait or a disease. How does this work? We typically don't use sequencing data, and so that's why we focus mainly on common small sequence variants, where you would have genotype at several places in the genome where you can have one letter or the other. And basically what you work on is a very large matrix with hundreds of thousands of participants in rows and 1 to 10 million genetic variants in columns. And then we count, in a sense, how often at a given position you have the alternative allele compared to the reference. The reference comes from the reference genome. It's not necessarily the most common allele. It's just an arbitrary choice. And so these genotypes look like this, that you have some points here in the genome where you know that there was one letter or the other. We do a bit of quality control, and then based on these letters, we can infer 99% of the genome. So how does this happen? We have a few places on the genome where we know that we have 0, 1, or 2 different alleles. And we compare these positions in the genome with known reference panels. These reference panels are people who have been sequenced with very high depths, where we know the full sequence. And the idea is very smart. It is that we inherit our genes, not one letter at a time, but in blocks. And in these blocks, the letters are correlated. So whole chunks of the genome where the letters are correlated. So you don't need to read each and every single letter. If you see that you have a bunch of letters on a given block where you know that they are correlated, you can infer the missing letters. These blocks of letters that are correlated, we call them haplotypes. And thanks to the non-random distribution of haplotypes, we can infer the missing points in our data. The fact that the alleles in the genome are correlated is something called linkage disequilibrium. Since we have the alleles that are not randomly distributed, they are not in equilibrium, we call that linkage disequilibrium. And this is a fundamental concept that we're going to use a lot today. So I said in our lab, we're very much interested in babies, and once they are born, the first thing that you do typically is you weight them. And so if you want to run your first US, this is really the one of the most standard analysis. You would look at all the different positions in the genomes where we have a change of letter, measured or imputed, and compare it with the weight of the baby. If you see no differences between different alleles, you would say that there is no effect. If you see that the more letters, the more alternative alleles you have, the heavier or the smaller the baby, you would say that there is an effect. So this is typical weight of babies in Norway, and typically you would adjust for things like batches, sex, gestational duration, etc., and just run a simple linear model with a few tweaks. The power line that we were looking at before, you see, is looking exactly the same, that we have here our limit of significance, and here the SNPs that are showing out. And the difference with before is that here I'm plotting everything, so you see the SNPs that are coming out, but also all the ones that hitchhike with this same point. So the classical way to look at it, it's what we looked at for nausea and vomiting, where you plot all the different points on the genome and the significance of the association. And you see here something that is quite characteristic to genetic epidemiology is that so you do your standard experiment in the lab, you're happy if you have a p-value of 0.01. Here we are doing 10 million tests, so a p-value of 0.01 or even 10 minus 5, we would have tons of them. We essentially only look at things that are below 5, 10 minus 8 in terms of p-value. And there you see these large peaks that tell us, oh, okay, there is a gene around here. There is something where you change the letters, the babies get bigger or smaller. And if you zoom in, that's something, so when you look at the whole genome, we call that a Manhattan plot, because you see it looks like Manhattan skyline. And if you zoom in, you look on these peaks, you see these clouds here. And so these clouds is what I was referring to before. The letters are correlated. So when you run a test, all the letters that are correlated are going to show some signal around the same position. And our job in genetic epidemiology is, in essence, to try and understand these peaks, look at how they link to the disease, how they link between diseases to inform on what drives traits and diseases in the general population. Now, our group, as you understand, we are very interested in proteomics, and recently there's been a lot of work on trying to add the proteome as an additional layer of information, try to better understand what these peaks want to tell us. Typically, you would want to know what proteins are in there and at what concentration. Once again, we are going to work on population level data, so we need rather large scale data, we rarely work with less than 100,000 or 1,000 samples. And to get samples at these scales, typically it's samples that are stored in biobanks from large cohorts. So that would be biofluid, most of the time plasma or serum. 3 different technologies are mainly on the radar these days. Antibody or affinity assays that will try to bind specific proteins and give you an idea of the protein abundance. It has rather low specificity and accuracy, but it's something that you can run up to millions of samples in large biobanks. It just gives you a table. For this protein, we think that we have this intensity. For this protein, we think that we have this abundance. Liquid chromatography coupled to mass spectrometry, I think that is what you have been most looking at in this course. It's something that will give you higher sensitivity and accuracy, but in terms of running hundreds of thousands of samples, we rapidly hit a bottleneck, either in sample acquisition or in data interpretation. The thing that is interesting is that once you have measured your proteins or you measure peptides, you can get some sequence level information. That's something that in my group we're very interested in. In the future, there is now more and more hope that we will be able to sequence proteins. There are a few technologies that are appearing, notably sequencing using nanopores that is hopefully going to hit the market in a few years' time. But it's still experimental. I have never seen large-scale data on this. How do these platforms compare? You have tons of paper on this. The short answer is not very good. Even within the same manufacturer, you use different technologies, different batches. It does not work well. But as we do in genetic epidemiology, look at many different samples. At the end of days, with thousands, tens of thousands of samples, the signals tend to make sense, and the biology of the data converges. A few things that you might want to be mindful is that each technology has its own proteins which performs better than others. You have some blind spot on some biases in others. And here is an example where people compared how much we can correlate proteins with known genetic variants that would affect the abundance of the protein. And you see that in these cases, for example, the affinity-based approaches would tend to miss or to favor different types of the same proteins. We call these proteoforms. But once again, once you run it on very large cohorts, you get results that you project a lot of the noise, and you get results that are significant at a population scale. The most advanced resource in this field is from the UK Biobank, where they have processed thousands and thousands of samples, and now they want to run, I think, 500,000 samples from more than 500,000 samples on this cohort. And from there, you can start combining diseases with proteomics at scale with genotypes. And a flagship example is looking at when you have a variant that is associated with a change in the protein. This is something that we, in the protein abundance, that's something that we called a protein quantitative trait loci, PQTL. And you see here on the position on the genome in red some spots where a change in the genome would change the abundance of the protein that is really nearby your variants. That's something called a cis-QTL. And in blue, variants where you would change the genome, and it affects a protein that is really far away. You see, it can even be on another chromosome. When I say it changes the protein, I mean it associates with a change in abundance, and this is called a trans-QTL. And if you do the same exercise as before and look at the significance of your association over the genome for a given protein or a given set of proteins, you would see things that genetically that are in a way coordinated in their association with the genome, which gives us a hint that, okay, if these proteins somehow move in the same direction when you change the genome, most likely they are doing something together, and there is some kind of biology constraining the stoichiometry of your protein. And there you can understand that these proteins would be important when they work together. And if we see that this change is linked to a disease, this process would be critical for a disease. This is something where you will find tons of papers these days trying to understand, okay, if we have these genes and proteins levels that are correlated and moving in the same direction, what does it mean for our disease? Another thing that where the proteome can inform your epidemiology approach, and that's data from our lab. We looked at the patterns in pediatric diabetes in Norway and saw that if you just look at the clinical variables, the children, they are all a little bit different. It's not that they typically children you would say, oh, it's type 1 diabetes or early type 2 diabetes or monogenic, but even within these classes, you have some substructure. And the question was, yes, but what is driving this substructure? Is it something biological behind it? And there has been some discussion that you would have two different endotypes in, especially in type 1 diabetes, where that show different patterns in the pancreas. Now, these children, they are alive, happy in Norway. We have no way to investigate the pancreas. So instead, what we did, we had genotypes on all of them, and we took blood samples on a few hundreds, and look at at a set of NMR panels in terms of very different indicate markers of lipid metabolism like cholesterol and also proteomics. And from there, we could see in the proteomic signatures in blood that indeed the different patterns that we saw in childhood diabetes were aligning with what our colleagues saw in the dish when working on beta cells. And that is a strong marker for us that, okay, this pattern that we see on the clinical side is mirroring what people would expect in the lab, and that we are indeed looking at two endotypes of diabetes. And from there, once we understood, okay, this pattern in our data is not just driven by age, but it's driven by the biology of the disease, our colleagues clinicians can take it forward to improve the treatment of these kids and to avoid that they develop complications later on. So far, we have been looking at proteins without really thinking too much what is a protein. And that is an error that we often do in genetic epidemiology. We think, oh, we have a variant affecting a gene that affects a protein. But a protein can be a very different beast, whether you look at the thing that is just expressed from your genome or the one that is expressed in the blood or the one that is actually doing its job. Here you have the example of insulin. This is the part that your beta cells produce in the pancreas. It gets really reworked before you have something that you can release in blood and that can do its work. And whether you measure one part of a protein or another part of a protein, that would give you completely different information. So in our lab, we try to go a little bit beyond this idea of one gene, one transcript, one protein, and try to understand a little bit the beautiful mechanics that are happening in the cells before we measure them in blood samples. Why do we do that? Here is again an example from the pregnancy paper I was talking about before. Where you remember when I told you there is this hormone when mothers feel it sometimes they get really sick. This hormone is produced by the pancreas, by the placenta during pregnancy, and is making the mother sick. So how come do we find signal only on the maternal side, or is there some signal on the fetal side? And this is a beautiful experiment that has been done in this paper led by our colleagues once again in the US and in the UK, where they looked at this hormone and they saw that some people at close to the N terminus carry an H or a D. And so they made an assay that could measure whether on your protein you have an H or a D. And then they correlated these abundance estimates with the genotypes of the mother and the child. And they saw indeed, oh, if the child has an H, the protein gets an H. If the child has a D, the protein gets a D. So clearly the protein is fetally expressed and not maternally expressed. Another thing that we are very much interested in is whenever you have a genetic signal here, it's another very interesting one. It's a set of genes in the fetus that produce proteins that we call pregnancy-specific glycoproteins, and these pregnancy-specific glycoproteins affect how much the mother will gain weight and how healthy she's going to be. You see, the genetic signal spans, I don't know, 20, 30 different genes. So how do you find out whether it's one or the other that is mediating the disease? These are things that we're currently investigating, trying to find out whenever we see a peptide, is it one PSG or another? Is it encoded from the fetal genome or the maternal genome? And how is it mediating maternal health in the beginning of pregnancy? And you see, if you want to understand what isoforms are we looking at, is it encoded on a fetal or the maternal genome? Just having one intensity per gene does not help us. We really want sequence level information. So how do you get sequence level information in proteomics? Most of your money is going to go in the sample preparation. Once again, we're looking at plasma or serum samples. So if you want good coverage of your proteins, you need to have very expensive sample preparation that would extract your proteins and try to make sure that you don't lose the low abundant proteins that are very insightful for us later on. The proteins get digested into smaller bits, a bit like you would do in NGS, and then you throw them at a liquid chromatography system that would spread your peptides so that they don't get analyzed all at the same time, and you in essentially break them into small pieces and measure the weights. On the other side, on your computer, you would take what you expect in terms of genome, predict what you expect in terms of spectra, and compare the two. One important idea here is that this comparison is a statistical process. So the more data you have, the more samples you have, the better your bioinformatic workflow or program is going to be able to extract signal from the noise. So long story short, you will find more proteins if you have 1,000 samples than if you have 100. Now, one thing that we noticed when we started this approach is that when we were doing genetic epidemiology, we were very careful. Okay, do we have the right genome, the right haplotype for the right person? I said before, we need to have the right reference so that we can infer the letters. But when we work with proteomics, we just take random sequences from UniProt. Where they are not random, they are derived from the reference genome. But that means that, in a sense, our proteomic approach is completely blind to all the variation that were key for us to understand the biology of diseases in genetic epidemiology. So it's called the streetlight effect. You take a database of the reference genome, you find reference proteins. Everything that is outside this, you are blind to it. And so Jakub, who you see here working in the lab, went back to the reference panels, and just like you would do in a genetic epidemiology approach, derived protein sequences that you could find from these reference panels. He wrote a tool that can do that for you, and he also made databases generated from public reference panels online for you to use. And surprise, surprise, the first thing that you see is that the moment you go outside the reference panel, you cover much better the different populations that you can find in the world, and you have much less bias towards different populations. Most of the so you would have much more genetic variation for people who self-identify as African. They have been through less bottlenecks in terms of population genetics. And if you go beyond the reference panel, you cover, for example, these samples much better. And so then we went into our spectra and tried to find out whether we could identify these haplotypes. So remember, these haplotypes are these stretches of sequences where you can have different sequence variants. And indeed, if you start processing your data with these databases that cover more the variety of who we can be, what kind of proteins can be encoded, you find many more peptides, but you find, in a way, too many of them. And that's something that we were expecting, but not to this scale. Even if you select the best peptides, the best peptide to spectrum matches, you would have approximately 1/3 of your results where a change of letter, where basically the computer cannot distinguish a change of letter from a post-translational modification or a modification that occurs during sample preparation. And so here, you would have, for example, a modification here that makes your match undistinguishable from a change of amino acid. I'm not sure whether it's the right example, but you get the idea. Proteins can be modified, whether in the cell or during sample preparation. And our tools are simply not good to distinguish these small modifications from genetic variation. So to try and see whether we could do something about it, Daphne, who is here and was working on this, has developed an approach where she would make the standard database search where you look for peptides and in your database with an open modification search, and then make the two compete so that you can be sure that whenever you find a sequence variant, you are not being fooled by a post-translational modification. And then that's something that I really like. She went into the results of this paper. You can get the data online. And just using the spectra, try to infer whether a person was carrying one allele or the other. And from there, we could reuse, it's a small data set, it's just, it's less than 100 samples, but from there, we could always see some pattern that how often you detect an amino acid versus the other, so how often you detect the product of an allele versus the other. We compared this with, so that's the proteomic frequency if you want, we compared this to the genetic frequency, and you see that you have some very rare alleles that you detect almost all the time. So this would be typically an error in your algorithm that systematically catches the wrong peptide. You have some alleles that are very common, but yet that you detect very rarely, and that would be typically that your instrument did not catch it, or that your computer did not manage to match it. But for the let's say half of your alleles, you would have a very good correspondence between the frequency in genetics versus what we see in proteomics. There is something very beautiful in genomics that we call the Hardy-Weinberg equilibrium, which means that if you have a good distribution of the alleles in the population, whenever they are transmitted to the next generation, it creates an equilibrium in terms of how many people can have two of the same allele, one of each, or two of the other. So these would be called homozygotes of the reference allele, heterozygotes, and homozygotes of the alternative allele. And you see here the Hardy-Weinberg equilibrium computed not based on the genetic frequency of alleles, but the alleles present around the diagonal here. So you see that you can infer the genotype of your patients for quite a few variants very reliably, and you can use the information from genetic epidemiology to find, okay, my results are off in terms of frequency or in terms of Hardy-Weinberg equilibrium. In a way, you can quality control your proteogenomic data in the same way that you would do in a GWAS pipeline. Building on this, Jakub, who you saw before in the lab, built web interface that is called the ProHub Explorer. And if you check his paper, he's presenting it, I think, this week or next week at the conference in data visualization. You basically type the name of your favorite gene, so here HNF1 alpha, and it will tell you when Daphne reprocessed these papers, we found it's mainly in the liver. That makes sense, in the intestines. And these are the different upload types that we expect, how much is covered by proteomics and the different peptides. And that, in a way, allows you to dig into proteomic public data in the way that you would do in genetic epidemiology. One thing that is really nice with proteomics is that we can also see things that are not necessarily encoded in the genome. So I was talking before about modifications, and in blood samples, we have tons of antibody peptides, and Daphne did some work also on how to map these changing amino acids to different antibodies and especially to variable parts of antibodies where we would not have the genetic information to infer this. It's still very experimental, but if you're interested in this type of topics, let me know. And so now we have a better way to design our expected proteome. And we have some better tools to understand whether when we make a match between variant peptides and these haplotype databases, how do we take it to a framework that would be like genetic epidemiology? So this is still very early data, but we are working on extracting data, extracting intensities for all these peptides. And so you see here, in a sense, how strongly the peptide is coming out and how well it can be identified in green. And typically in proteomics, you would focus only on the high quality data where you know I'm 99%, more than 99% sure that this is not a random match between my intensity and my sequence. But you understood in genetic epidemiology, we're not necessarily interested in having a perfect match in the sense that we have thousands of samples, and we want some. Even if we have some noise, even if there is some randomness in there, we want to keep the signal, because once we are going to analyze it over hundreds or thousands of samples, the signal will survive. And so we are now developing tools that will extract these noisy signals. That's the red and orange signals that are low in intensity, that are very variable. And we are correlating this with our genotype. And once again, common genotype, so we don't expect that the variant affects a lot your how your protein is expressed or how it's folded. And so if you don't have the allele that encodes the blue peptide, you would, if you measure something, in essence, it's noise. And if you have one allele or two alleles encoded in the peptide in blue under an additive model, you would expect a doubling of the intensity if you want. And for the green peptides, if you have two alleles, one allele, zero allele, kind of the same approach once you have scaled everything nicely. This is the theory. How does it look in practice? When you dig out these noise intensities, it's not too bad actually. And you see, this is just 100 peptides, and we already see a trend that whenever your patient encodes these specific alleles, we see the peptides carrying some intensity, whereas here, both for this expected relationship for the reference peptide and the alternative peptide. And that's something that we are heavily working on, how to interpret the proteomic intensities that we measure in the mass spec with the genotypes, and then link this to diseases. And with that, we have reached how far we have come on this topic. I hope that we will have more results to show you in the future in terms of how this links to different diseases. This is an outdated picture of our group. You see the beautiful mountains of Norway. Someone who is missing here is Anya Palaka, who came actually from the Kiev School of Economics just at the beginning of the war. Came to find shelter in Norway. See, she's here, and after some time started a PhD and is now very happy in Norway. So don't be afraid of the bad weather and the cold. If you're interested in, for example, having an internship, if you're interested in this type of research, would be very happy to welcome Ukrainian students or guest researchers. Thank you for your attention.
Hello. I wanted to ask if you have encountered alternative splicing in your work and how did you deal with it?
A very good question. We did not get. I took a few shortcuts. We tried to find which transcript would be mainly driving the association. We try to find peptides that overlap with splice junctions, but right now we are mainly focusing on trying to find the product of genetic variation. That's hard enough. I hope that we can get splicing variation at a later stage.
Thank you.
Yeah, Hardy, you can go on.
Hello, Mark. Thank you very much for a very exciting lecture. I have several questions. Maybe first a brief one on the methodology. How do you generate mass spectrometry reference databases for proteins when you have multiple variants that could be co-detected in the vicinity in the same peptide? And do you suffer from the database inflation in terms of FDR control?
So let me take the last one first. The good thing is that these common variants, so the first thing is that we don't have this combinatorial explosion that we need to look at all possible combinations, in the sense that we are building haplotypes directly from the genome. And there are not that many coding variants in the genome. So the database inflation is not too bad compared to, for example, what you get in proteogenic genomic databases that are produced from cancer samples, where you would do the full 6' translation. There you have like tons and tons of novel peptides. Here we're just tweaking a letter here and there. Then or a letter or a set of letters. And then this goes all into predictors for retention time fragmentation. And that's the part where you're right, we get problem that peptides can get co-fragmented, and sometimes really you look at a peptide with a variant and another peptide. Typically a problem would be like trioxidation of tryptophan, completely identical, even in the predictor. And that's the part where we take advantage that we have genetic information, because the co-eluting peptide will co-elute of your peptide independently of your genome, but the peptide that carries the product of your allele would be absent if you don't have the allele. You would have one version if you have one allele, or two versions if you have two alleles. And so it's the correlation gets, if you want, projected out once you correlate with your genome.
Okay, yeah, thank you very much. My question was indeed a bit inspired by the work in the cancer cell lines. So, but good for you that in your context it's a bit easier.
That's
regarding the abundance. So you mentioned this part about protein variants that are affecting the detection in the affinity-driven methods, and among other things, you had ELISA. We work with ELISA in the clinical setting, and we also do meshed RNA sequencing on the same samples. So since this bias exists, it would be great to be able to correct for this, right? In case we are nullizing a protein which has a variant in a given patient. So my question would be, are there maybe databases that we could use to reference against to try to infer the correction factors for the known things? Or would you advise not doing this because we don't have enough data, or trying to build something ourselves?
It's very good points. I am not aware of any database on this. What you can do is check where your antibody is binding and where your variants are located, and that's something that the manufacturer should check. If you ask me, that's like for GDF15, if you have a change close to the N-terminus, you don't want to take an antibody that will have its epitope here you want to target another portion of the protein. So if you have documentation on where your antibody is binding, you can check in public databases like UniProt, do we have common variants here, or do we have variants that are commonly found in my cell lines on this? But yeah, antibodies, there are many problems in terms of sensitivity and so yeah, sequence variation is only one of them.
Yeah, okay, so because I was maybe a bit driven by the slide that you had that was cross-comparing different mass spectrometry methods and different affinity methods, and I was wondering if those estimates could be used to learn correction weights.
Oh, that's a very good idea. I'm not aware of anyone who tried, so go for it.
All right, thank you very much. Thanks again for the lecture.
I will read some questions from the chat. I'll go from the last one. So Marina thanks for the lecture and asks, according to your research, what influences vomiting during pregnancy more, the mother's genes or fetus genes?
Aha, very good question. What do you think? We can make a vote.
Okay, who is for mother genes? Put your hand up.
Do we have smileys? You can put a heart for the mother and a party sign for the baby in the smileys.
It looks like mother wins, both by
yes,
smileys
exactly.
and hands.
Mother wins, yes. It's really the sensitivity to the hormone that is driving the sickness more than how much the baby produces. So if the baby produces more, you have higher chances of being sick. But it's really if the mother has a high sensitivity to the hormone. And the researchers in this paper have shown that if the mother was exposed to the hormone before pregnancy, then she's less likely to be sick. So now there are trials running in the US, in the UK, one soon starting in the Netherlands, and we're looking for funding in Norway to start one, where you expose mothers who are at risk before the pregnancy so that they get used to the hormone, and we hope that they will be less sick afterwards. It's a very good question. Yeah, it's mainly maternal sensitivity.
Thank you. Also, someone is we have someone that can unmute themselves. Yes, Anha?
Yes, thank you for the lecture a lot. And I want to ask about counting the missing values, because as you mentioned, you usually work with very large cohorts, and I'm really interested in how you distinguish between the proteins that varies through the population and the proteins that are just low abundant, so you don't get them in all samples, even if they are in them.
Yeah, that's a big problem for us. So we try to get intensity as much as we can, and then it's really try to get more samples. So I prefer to have low intensity that is that still carries some signal over many samples than a missing value. The missing values, there is not much we can do about it. We tried different models to impute the missing values. It's never working very well. So now we try to dig deeper in the samples and get more samples. Sorry, it's maybe not a good answer.
No, it's good. It's pretty good. Thank you.
Right, as you said, the more samples you have, more proteins you get. This is an advantage probably of the large-scale proteomics analysis, but also there should be a lot of difficulties like, computational efficiency, like you need huge servers and grad students who can do all of that.
Yes, and of course, all these data are sensitive, so you cannot just put them on Google Cloud or on Amazon. I see that there was a question: which language do we usually use? In our lab, it's a bit of a mix of many things. When we need something handling large scale or to run quick, we write it in C++ or in Java. So the last work that I was showing with the intensities that we grab from the low abundant proteins, it's all C++. And afterwards, once we have extracted the intensities or done the analysis, all the post-processing, like statistical work, happens in Python or R. But if you ask two people in my lab what is the best language, they will never agree. So that's okay.
That is really nice to know, because usually, at least people who start with biological backgrounds, they first learn Python and R, and many stop there, but probably if you want to do fast applications, then you need to expand your toolkit.
It depends on how deep you want to go in the data, I would say. If you really want to go to the raw data of 1,000 samples, if you want to touch really the low level intensity, then C++ or Java are hard to beat. Yeah.
I see. Thank you. And we can conclude with a last question from chat. And Natalia asks you to elaborate about internship opportunities.
Yes, so we often welcome students who want to have internships, or can be a few months on our whole master's project. There are different ways to fund these, from if you get funding from your home country, or we can sometimes get funding from the EU, depending on whether you're eligible, or from Norway. And then we can build us a project for you to come and work with us a few months. And so that's typically for students who are doing their masters and who want to work on a project with us. If you are, say, at the PhD stage or later, we often welcome researchers who come for a visit a few months or up to a year. So of course, we often advertising positions. So if you're interested in these things, just let me know.
And I remember you mentioned about remote internships too. Is that still true?
Yes, that depends. It's always a bit more difficult to organize in the sense that we like to be in the same place to discuss things. And many funders would not fund remote internships, so that and the money can become a problem. But on my side, we're always happy to have remote students who want to work on a project, and we're happy to welcome them.
Thank you. It's something that is common in Ukrainian universities, that we have like a practical session whenever the student find themselves in the laboratory to spend 2 to 3 months of full research, and it can be either in Ukraine or abroad. So maybe this is something that our audience can consider.
Yes, we have the same thing. That would be compatible, and I'm sure we can work out if you need credits or something that it gets approved.
That's perfect. Thank you very much, Marc, for your time and sharing interesting research. And we'll
Thank you, and thank you for the nice questions.
Thanks. Then everyone have a nice evening.
Thank you, bye.